Hi everyone! For those of you who I have not had the chance to meet, my name is Alicia and I'm a first-year PhD student in evolutionary anthropology, and I will be guest lecturing for EVANTH101 today with Maddie, who is another PhD student in the department, and she will be lecturing in one of the videos later on in the lesson. Today we're going to run through a few of the basics of functional morphology, but before we get started, I'd like to outline a few of our learning objectives. By the end of this lecture and review of the material, everyone should be able to do the following. First, recognize and name the different bones of the skeleton. Define functional morphology and explain its application to the study of human evolution. Identify the skeletal correlates of different primate locomotor repertoires, and identify skeletal correlates of different primate dietary repertoires. Now, let's get into the first portion of the lesson. First, we're going to run through a sort of crash course on the skeleton and anatomy so that we can all be on the same page when it comes to talking about how we go about studying the skeleton. Here, we're going to be working towards our first learning objective, to be able to recognize and name the different bones of the skeleton. So what is the skeleton for? We all know we have one and that it literally is the backbone to us being able to walk around and function day to day. But what key purposes does it serve? It provides the body with structural support, protects sensitive organs, so think about how your ribs create a barrier between the outside world and all of your organs, allows for mineral storage, promotes immune function by producing immune cells, and provides leverage for muscles to act on to enable movement. That last point is what we're really going to focus on during this lecture. The skeleton provides leverage for muscles to act on to enable movement. In studying the skeleton, we can learn about how different animals move. Now, there are 206 bones in the body, which can be a sort of intimidating number. So in order to better study the skeleton, we often break it up into a few different functional regions. The forelimb, which is your arms, the hind limb, which is your legs, and the axial skeleton, which is your head and kind of the bulk of your body. So to make this a bit more digestible, we're going to go region by region. First, we're going to talk about the forelimb. Within the forelimb, we have three main functional units: the upper arm, forearm, and then the wrist and the hand. In the upper arm, we have the humerus. This bone connects to the rest of your body at your shoulder. There's a big round head at the top of the bone, and that allows you to rotate your arm around. Moving down to the forearm, we have two bones that work together to allow us to twist and rotate the arm. These are the radius and ulna. A perfect example of how these two bones work in tandem is if you hold your arm straight out in front of you with your palm facing down. Then slowly twist your arm so that the palm is facing up. In doing this movement, the radius and ulna cross over one another, which allows you to move your arm in the way that you just did. Then, at the bottom of the radius and ulna, we have the small, tightly packed bones of the wrist. You don't need to worry about these bones, as a lot of experts struggle to name and identify all of them. These are called the carpals. The human wrist and hand itself is made up of 27 bones. That includes the bones of the wrist, as well as the long bones of the hand that make up the palm and fingers, or digits. So now let's review the hind limb, aka your legs. First, the hind limb begins with your pelvis, which is what makes up your hip bones. If you press on your hip and you feel something hard, you're touching your pelvis. The femur, or the thigh bone, connects to the pelvis, similar to the humerus in the forelimb. The femur also has a round head that allows for increased range of motion at the hip. This is why we're able to walk and run and jump and do splits. As we move down the leg and reach the bottom end of the femur, we run into the tibia and fibula. This is another two-bone pair, just like we see in the forelimb, but these bones don't cross over one another the way the radius and ulna do. If our tibia and fibula could freely rotate the way the bones of the upper limb do, we likely wouldn't be very effective walkers. So the tibia and fibula stay securely in place so that we have more stability in the leg. At the very end of the tibia and fibula, we are met once again with a group of small, tightly packed bones. These are called the tarsals, and they make up the ankle and the hind foot, or that back portion of the foot. You also do not need to learn all of these bones for the same reason you do not need to learn all of the carpals, the small bones of the wrist. Then, as we move into the foot, we have the metatarsals and phalanges. These make up the bulk of the foot and the toes. Then finally, the last functional region of the skeleton is the axial skeleton. The axial skeleton forms the long axis of the body and is made up of the skull, which is your head, the vertebral column, which is the neck and back, and the thoracic cage, which is the rib cage. The elements of the axial skeleton are highlighted in this image in blue. These are core anatomical structures that protect your vital organs, like your brain and your lungs. So now that we've learned about the functional regions of the skeleton, how do we actually study skeletons? How do we make judgments about bones without just making things up? Well, we do this by studying comparative anatomy. Just like how you approach problem solving in many different fields, we have to compare skeletons to one another to make meaningful judgments about their function. Everything is relative, in general, but especially in comparative anatomy. When discussing primates and animals in general, we must keep in mind the variation in anatomy that is present among different taxa. On this slide, I've added a couple of examples of how anatomy varies between different genera. On the next two slides, we're going to explore these differences a little bit more so that everyone can get the hang of making anatomical comparisons. So, for our first example, these images are all 3D renderings of different feet. From left to right, we have a modern human, chimpanzee, gorilla, and orangutan. At first glance, I'm sure many of you are noticing a lot of variation between these 4 feet, and you don't have to be a well-seasoned anatomist to see these key distinctions, so let's run through a few of the bigger ones. First, there's a difference in the size of the hallux, that's the big toe. In the modern human, the first bone of the big toe is really robust. I've marked this with a red line. When you compare it to the size of the hallux of the orangutan, which is the 4th foot on the screen, there's a big size difference. Next, you may have noticed that the modern human foot has a hallux in line with the rest of the foot bones. What I mean by this is that the big toe is fixed in place like the other toes, and the other examples of the other feet on the screen, you'll see that the hallux is actually abducted, abducted, meaning that it is separate from the other toes. Next, the overall length of the digits is different among these feet. The orangutan, for example, has really long digits when you compare it to the other individuals pictured here. Now, let's do this again, but with the axial skeleton and pelvis. Here, we have a gibbon, which is a type of lesser ape, a human, and a chimpanzee. Just like in the last example, I'm sure you all are noticing some differences between these 3 individuals right away. If you'd like to challenge yourself, pause the video here and note 3 of the biggest differences you see between these 3 individuals, then unpause and see if you notice some of the features I'm going to talk about. You can pause the video now if you're choosing to do so. Okay, so the first difference is the height of the pelvis, or the hip bones. You can see in the gibbon and in the chimpanzee that they both have pelvises that are way taller than the pelvis of modern humans. Second is the width of the pelvis. The modern human pelvis is wider than the gibbon pelvis. Third, the overall shape of the ribs is different among these different individuals. For example, the chimpanzee rib cage is triangular, much more so than the human or gibbon rib cage. Understanding differences like these ones that we just identified in the foot or in the axial skeleton and pelvis are key in making meaningful comparisons between and amongst different taxa. Now, regarding the fore and hind limb, we use something called the intermembral index to make judgments about how an organism might move. Among primates, we show a lot of diversity in the way that we move, and this is reflected in the lengths of the arms and the legs relative to one another. To calculate the intermembral index, we divide the length of the forearm by the length of the hind limb, then multiply by 100. This gives us a number that we can use to learn about the different locomotor modes that different organisms use. Locomotion is essentially how an organism moves or walks. Suspensory primates use their arms a lot, and because of this, tend to have relatively long arms. Quadrupeds use both their fore and hind limbs a lot, which means they tend to be around the same length. Then bipeds, like humans, rely more on our legs than on our arms, and so we have relatively long legs when compared to our arms. And finally, the last bit of information we're going to touch on in this section of the lesson is differentiating between shared function or shared ancestry. This can be really complex and still confuses researchers every day. Earlier in this lecture, we reviewed the bones of the forelimb in humans. You'll notice in this image that a lot of other organisms have strikingly similar bony structures in their forelimb. But does this mean that we share a recent common ancestor? No. Sometimes different structures are similar to one another because the organisms we're comparing may use their bodies in similar ways. These are key distinctions that researchers have to make when studying comparative morphology. Finally, we're going to go over a couple of key terms to keep in mind when asking important evolutionary questions using these methods. First, we can all agree that the images on the right side of the screen are homologous structures. They're all the same or very similar. These structures that are similar but appear in different organisms are also referred to as analogous structures. But because these similarities are not a result of common ancestry, they're examples of homoplasy. This is exactly where a lot of researchers make mistakes in trying to make taxonomic claims. If a trait is a homoplasy, then the two similar structures are a result of convergent evolution, meaning that these two similar traits evolved independently. If you think back to the rock pocket mice, these were an example of convergent evolution because they all evolved dark fur independently. So now, after the first portion of this lesson, you can all recognize and name different bones of the skeleton, as well as begin to understand how we compare the bones of different taxa. See you all in the next video.